Hello again, and moving on from DNA replication, we come to PCR, or polymerase chain reaction. Okay, so what is PCR? It's used to amplify or to make many copies of DNA in vitro, which is outside the body in lab conditions, and it has lots and lots of different applications. So, what are these applications? Well, firstly, it can be used for genetic mapping studies like the Human Genome Project. It can also be used for forensic testing, which is sometimes called DNA fingerprinting, but this is just an analogy, as fingerprints are unique to a person, so is their DNA. It can be used for parental testing to see who your parents are. It can be used to classify a species, in which you compare the genomes to find the degree of relatedness between them, and we see this again later on in this unit. And it can also be used to screen for and diagnose genetic disorders like cystic fibrosis. An important part in the development of PCR was to find heat-tolerant DNA polymerase, as the first stage involves temperatures near the boiling point of water. Now, the DNA polymerase used was found in the archaic bacterium Thermus aquaticus, which lives naturally in hot volcanic springs. This is why the DNA polymerase is often described as Taq polymerase, Taq being TAQ. Now, if we quickly have a look at the summary of PCR, what you begin with is a supply of your target DNA, that which you want to copy, the Taq DNA polymerase, primers, and nucleotides. The first stage, the reaction mixture is heated to 94 to 96 degrees centigrade, and the DNA strands denature or separate. For stage 2, the temperature drops to about 50 to 65 degrees C, and at this point the primers attach to the DNA strands, and this is called the annealing of primers. For the third stage, the temperature is cranked up again to about 72 degrees C, and it's at this point that the DNA polymerase replicates the DNA. And this process is repeated over about 20 to 30 cycles as necessary. Now, you can do PCR in the laboratory with water baths at different temperatures and a stopwatch, but nowadays it's done automatically using a thermal cycler. So what are the full list of things that you need for PCR? Well, firstly, you need the target DNA to be copied. You need your two types of primer. You need a supply of your enzyme, your DNA or Taq polymerase. You need a supply of ATP to provide the energy for stitching all those nucleotides together. And of course you need a thermal cycler machine. So let's have a quick overview of PCR. Firstly, we start off with our strands of DNA containing our region of interest, or our target DNA. So we begin with stage 1, where the temperature is increased and our DNA strands separate. Now, even though DNA replication is taking place, there is no replication fork to open up. There are no leading or lagging strands. When the strands denature or separate, they separate along their entire length. So at stage 2, the temperature drops and the primers attach. This is called the annealing of primers, and there is a different primer for each strand. Stage 3, and this is where the DNA replication takes place. The temperature is cranked up to about 72 degrees C, and the DNA polymerase comes along. Now, you already know that DNA polymerase can only add nucleotides to the 3' end of a growing DNA strand. So this top DNA polymerase molecule will work its way off to the right, and the one on the bottom, as you can see it now, works its way off to the left like so. So at the end of stage 1, DNA replication is complete, and where we had one complete double strand of target DNA, our region of interest, now we have 2. So our DNA made by the first cycle now comes to the beginning of the second cycle. Stage 1 occurs where the DNA denatures and the strands separate. The annealing of primers takes place and the DNA is then replicated. So this part of the picture here, that would represent stage 1, and this bottom part of the diagram here would be 2 plus 3, stages 2 and 3. And this is similar to the handout you may be getting in class. So, after the second cycle comes the third cycle. Heating of the strands again occurs and they once again separate or denature. Annealing of the primers takes place again, and so does DNA replication. So with this third cycle, that is stage 1 and that is stage 2 and 3 together. But it's only at the end of the third cycle do you get your copies of target DNA on its own being produced, and they are there and there. In subsequent cycles you just get more and more copies of target DNA being produced. Now in this amplification graph that you can see, it shows that with only 30 cycles of PCR you can produce 1 billion copies of your target DNA. So to finish off, if you want to see a much better amplification animations, etc., then type in your Google PCR animation and you can check out this website here. And when you come to the site, you can check out the animation like I've just said by clicking that link there, or you can see a better version of the amplification graph like so. Okay, thank you very much for your attention once again. Bye bye.